Hi everyone, my name's Steve Locke and I'm actually going to be replacing Suki today. Sadly can't make the presentation, so I'll be taking his place and talking about how you can use CE to analyze RNA and DNA molecules. Just give me a few minutes and I will share my screen. So this talk today is to discuss and to cover how CE can be used for RNA and DNA analysis, and how this is enabling the development of the latest cell and gene therapy drugs and vaccines. This has been really springboarded in development since the COVID outbreak, as CE has been used quite extensively to help the quality control of vaccines and also into the research of LNP and viral vector type delivery systems for the next generation of drugs. So just want to go through some of the contents that I'll cover today. So let's go back a slide. So we'll cover some background to the use of RNA in cell and gene therapy. We'll look at how you can use CE in developing methods for oligos, including small guide RNA, ASOs, and small interfering RNA. We'll try to discuss how you can develop methods for larger mRNA or messenger RNA targets. We will look at how CE has been used to analyze plasmids and DNA molecules, and then we'll finish up with a summary of the content today. So as I mentioned, we'll start off by a bit of the background in the use of RNA and DNA in cell and gene therapy. I think this is really an exciting space, and really this slide just covers one of the reasons why you target RNA. The conventional view of RNA is that it transfers information, but it also turns genes on and off. It aids chemical reactions. It alters other RNA molecules and helps build proteins. So you could actually, if you think about using RNA, it really is the part of the messenger of the body system to eventually build proteins, for example. And this schematic just shows you where some of the RNA is involved. And you can see here RNA is involved in transcription, translation, and reverse transcription in the conversion of messages to eventually change how a cell works. So you can see how RNA can be used to help fix a broken biological process. Now, DNA and RNA in cell and gene therapies really span quite a wide area of biopharmaceuticals. You can start off with really small chunks of information, for example, oligotherapeutics, ASOs, antisense oligonucleotides, siRNAs, or small interfering RNA molecules. And then you can move into small guide RNA and Cas9 mRNA. This is part of the CRISPR therapy system. Then you look at, go back a slide. Then you can look at the prime editing guide RNA, and then you can move on to mRNA size assessment. And this is really for your LNP vaccines or lipid nanoparticle vaccines. And really, the end bit, you're looking at plasmids. And plasmids really are used to help produce viral vectors, but also can be used to produce mRNA itself. So this is a sort of an area where you can consider these biopharmaceuticals, which are using DNA and RNA. And all these areas can involve the use of capillary electrophoresis, or CE. So RNA therapies can be sorted into one of three broad categories. Therapies that target nucleic acids, either DNA or RNA, therapies that target proteins and those that encode proteins, or hybrid approaches that combine several RNA-based mechanisms into a single package. And these are also now emerging as the more recent flavor of RNA-DNA biopharmaceuticals. Oops. Go back to the next slide. So the types of RNA therapies are listed on this page, and I think if you look at ASOs, they're probably the most common therapy which has undergone FDA approval, and they're really small 13 to 25 nucleotides in length, and they really prevent mRNA from working. So they're like a blocker, and this stops mRNA coding and help building proteins in the cell. They can also alter splicing and stopping a precursor mRNA become inactive. So they sort of interfere with the mechanisms of protein generation in cells. The next class is siRNAs. These degrade mRNAs, preventing it again being translated into target proteins. They're similar sort of length, but slightly different action to ASOs. RNA aptamers, another class, not as common as the first two classes, but these bind to specific targets like proteins and can help in cell delivery of siRNAs. These are generally a lot bigger, 50 to 120 nucleotides in length. And then, of course, the biggest of these types of RNA therapies is messenger RNA, used in vaccines, and these are thousands of bases long. And I think one of the most interesting areas in this space is CRISPR, and this really is gene editing. And gene editing, and it really uses a biomolecular technology to permanently change the genomic DNA information in a cell. CRISPR stands for clustered, regular, interspaced, short palindromic repeats, and it offers a fast, reliable, and precise approach compared to some of the legacy approaches for gene editing. The critical components of this approach are really the guide RNA and the enzymes. The guide RNA actually directs the enzymes to the portion of DNA which is going to be edited, and they form a ribonucleoprotein complex. CRISPR-Cas-dependent gene editing can be adapted to a wide range of applications, and the first drug in this particular space was just recently FDA approved. So I think that was from Vertex Pharmaceuticals. So it's a brand new space of biopharmaceutical drug development, and even there's now recently been a drug approved in this space. So types of guide RNA molecule used. Now, guide RNA molecules enables the CRISPR protein complex to target and cut a specific sequence in a genome. So basically, what you're doing is you're targeting these molecular scissors to chop out maybe a faulty section of genetic information. And the guide RNA can be separated into two pieces for easier manufacturing, or produces a single guide RNA sequence. There's the next generation guide RNAs, and these are prime editing guide RNAs. And these are slightly bigger, but they can enable editing with improved precision. So again, this space is continually evolving. And this gives you some idea of how these synthetic PEG RNA oligos work for prime editing. I won't go into too much detail, but it just shows you, this slide gives you some information on about how they're used. And again, these are quite long in length, and they can be up to in excess of 100 bases long. So not the size of messenger RNA, but still longer than ASOs or siRNAs. Now, current analytical methods are unable to accurately determine nucleic acid purity for complex, longer lengths like PEG or prime editing guide RNA. Prime editing technology relies on complementary sequences, design of the PEG RNA to enable unique functions. And CGE, or capillary gel electrophoresis, offers high resolution analysis of the main product from closely related impurities. And it's a size-based approach to really separate the prime main peak from size-based impurities. And it's a technology now which has been utilized in the quality control of this type of RNA. So looking at the next section, we look at developing methods for this oligonucleotide class, including sgRNA, ASOs, and small interfering RNA. In this has been around for a while, because ASOs as a biotherapeutic have been around for a number of years now, and as FDA-approved drugs. And it uses capillary gel electrophoresis as a way, again, to profile size impurities of these smaller oligonucleotides. What we do is we use a gel as a molecular sieving, a matrix, to enable the size separation of the main peak from maybe clipped variants, which are formed in the manufacturing process. One of the benefits of using CE or CGE in this approach is it actually has highly superior resolution to conventional slab gel technology, can enable quantitation and is highly reproducible and can be automated in operation. Comparing it to HPLC, again, it's more resolving than HPLC, and it's been demonstrated as effective and reliable in the biopharmaceutical area. Oops. And this slide here just shows you sort of how we work in this space for looking at these smaller oligonucleotides. In this case, we use the P800 Plus system as shown in the top. We have excuse me, ssDNA 100R kit, which is single-stranded DNA kit, which is used to look at smaller base lengths, so up to 100 mers. And you can see how on the far right-hand side of this slide how we can separate 40 to 60 bases with a single base pair resolution. And that's essentially what you need when you're looking at these ASOs material. This example here is taken from a Cytiva application, actually, where we've actually used this CGE separation using this ssDNA kit to look at compare crude samples with a product pool and to see if we can separate away non-tritulated failure sequences. And this is a good way of how you could use CE as an aid for process development. That was looking at a 25 mer oligonucleotide, so similar length to, say, an ASO. But again, this same kit can be used to look at guide RNA or slightly longer RNA than an ASO. So you're looking up to 100 bases. And again, when you each time you make this injection, of course, you replace the gel buffer. So the way the system works is that it's an empty capillary. You fill it with the gel, you inject your sample, then the sample is separated as it flows down this sort of gel matrix sieving, which acts like a sieve. And then afterwards, you actually clean out the whole capillary again and redo the process. And this really allows you to actually look at the synthesized products. Often these oligos are synthesized chemically, and to be able to separate the full length product from its lower molecular weight in process impurities. And we have a few application notes in this place. And if you're interested, I would advise anyone to go to the SCIEX website to look at the application notes for this particular area. But when you go to PEG RNA, actually, these are longer. These pegylated RNA, or sort of primatizing guide RNA, and these are longer in length, and they're longer than the classical guide RNA. And you can see here where we've used it's one of the attributes of our CE systems to help improve the separation. What we found when we first started to apply a classical method to analyzing this primatizing guide RNA was that we were getting broad peaks. The broad peaks themselves were partly down to sort of a refolding of the partial refolding of the RNA, which caused this bump. And what we did is we actually increased the temperature of the separation to improve this particular electrophoregram. And here you can see that what was causing one of the issues we were seeing was in this investigation on normal peak shape. So this primatizing guide RNA. These are, as I said, these are secondary structures forming, and you can see a couple of examples here from 129 ncRNA to comparing it to primatizing guide RNA. So non-complementary RNA was actually a nice sharp peak, but when you went to primatizing guide RNA, you get this broad peak, and that was caused by secondary structure formation. And then, as I mentioned, as we went to increasing the temperature separation, and actually we increased the temperature up to 50 degrees C, we now got sharp peaks like the ncRNA as shown in the previous slide. And one of the things you can do with SCIEX CE system is you can really control the temperature separation. So we actually went through a series of steps to change the temperature, and we ended up running the capillary temperature at 50 degrees C to remove this sort of structural interference that you were getting by looking at the PEG RNA, and this really enabled us to get this sort of sharper peaks under the denaturing state conditions. So we sort of unfolded the PEG RNA again and got it to produce classical sharp peaks. And again, one of the things we do in all our systems, not just the P800+, which is a classical CE system, but also the biophase, is that we can adjust the temperature, because we have an inert liquid that circulates around all the capillaries, which enables precise, stable temperature control during separation. On this slide, you're just showing you a pre-built cartridge, which is being used on the P800+ system. And again, once we sorted out the unfolding and the denaturing conditions needed for separation, the separation itself was very reproducible. And here you can see the RSDs and migration times for a number of injections. You can see it's down to below 2%, and the RSDs, the variability in the corrected peak area percentage, was again less than 2.5%. So at 129 base long PEG RNA, we're getting very reproducible analysis with the improved denaturing strategies that we used. One of the things you can do in order to speed up this type of separation as well is you can change the gel buffer, and that's something we also play with sometimes in separation. So you can use TBE to dilute the gel buffer to increase the speed. So that was looking at how you develop methods for smaller oligos, less than 200 bases in length. But how do you develop methods for larger mRNA targets? These are 1,000 bases now in length. And again, if you go back to the sort of CRISPR approach to biopharmaceutical development, the CRISPR is actually a complex. So it's a complex of small guide RNA, Cas9 enzymes, and mRNA. So it's a total complex, and sometimes you want to be able to measure the mRNA plus the small guide RNA in the same analysis. And to do that, you have to be clever in what you're doing with the gel you're using to do the separation. But in CGE, or capillary electrophoresis, you are able to actually look for both in the same analysis by just playing with the separation conditions. And here you can see an example of this. And here you can see Cas9 mRNA coming out at nearly 18 minutes, and the small guide RNA coming out with 100 bases at 9 minutes on the separation on a cartridge in CE. So here you can see a separation that's the red trace of widely differing sizes of RNA molecules. In the green trace behind it is basically a ladder, an RNA ladder, which helps us give size estimations for the so this sgRNA and the Cas9 mRNA complex. And again, to do this, we actually used a new kit. We used a different gel because we needed to change the gel to look for the larger mRNA. So we used an RNA 9000 purity and integrity kit, which had a new RNA gel. We actually apply this method not only to the P800+ system, which is a single capillary system, but we also use the same approach on the multi-capillary or biophase system, which runs basically 8 samples in parallel because it's based on a 96 well plate format. With regards to sample preparation, we try to simplify it. So we took our sample, we diluted it in sample dilution solvent in CE grade water. We then denatured it with heat using a thermocycler at 70 degrees C for 5 minutes. The samples then were cooled down before analysis on the CE system. And again, by using a kit either on the P800+ or on a biophase, you can get the same type of separations, which is highly reproducible. And actually, this RNA 9000 kit is actually now being used quite often by biopharmaceutical companies to profile messenger RNA and even single-stranded DNA found in viral vectors. The new gel, which is being used in RNA 9000 kit, has been designed to go for a range of 50 to 9,000 bases. So it's just not really designed to look at ASOs or siRNAs, the smaller ones, but to really look at sort of guide RNA length up to messenger RNA sizes of thousands of bases. The RNA gel is transferable between a pH 100+ and a biophase system. And the resolution is down to double-digit base differences at the high end in mRNA fragment analysis, and both in mRNA used in LNPs or lipid nanoparticles, but also mRNA found in the CRISPR-Cas9 complex. And this, what we do again in the system is we use laser-induced fluorescence as detection. Often, to be honest, when the concentrations of your RNA target drop low enough, you start to use dyes to tag your DNA RNA material, and when it's single-stranded, you tend to use like cyber green or cyber gold dyes to tag them. The tagging of the genetic material really enhances the sensitivity. And in this particular approach for mRNA, because often the concentration of the mRNA in these drugs is quite low, we tag it with cyber green. And here we see reproducible analysis of again the small guide RNA and the Cas9 mRNA, and you can see again it's really highly reproducible, actually for both, even on the same analysis in the same run. So we're seeing small guide RNA having a main product percentage CV area of around about 6%, and the Cas9 having a main product reproducible area of less than 2%. So again, CVs are very, very good. And again, this is simultaneous analysis of both small guide RNA and Cas mRNA of this CRISPR area in the same run. But I also mentioned mRNA can be used not together with small guide RNA as in CRISPR, but also used separately in lipid nanoparticles. And this is really a big area of development. And this is really springboarded on from what was used in COVID. Everyone's probably heard of lipid nanoparticles because we had them as a COVID vaccine, developed by companies like BioNTech, Moderna, etc., who developed this type of drug to treat the COVID disease, and we often use in COVID vaccines. So lipid nanoparticles themselves are like fat droplets which surround the mRNA, protecting it to allow it to reach the target cells. If you just give mRNA neat, like unprotected to a patient, the RNases in your own body will just break up the RNA, stop it delivering its message to the appropriate target cell group. So you need to protect it, and that's what the lipid nanoparticle does. However, to analyze it, you have to get the mRNA out of the lipid nanoparticle. So we use things like Triton X to disrupt the lipids and release the mRNA before you analyze it. And here is an example of mRNA vaccine, which is an empty particle, doesn't show anything, it's control, compared to an extracted sample which had a full particle. These results were run again using the RNA kit, which is used to run the Cas9 mRNA complex, and these results were actually run on our biophase multi-capillary system. And you can see on the far left-hand side the ladder, which has been used just to give us an idea of the sizing of the mRNA in this product. As I mentioned, use surfactants like Triton X extraction to break up the LNP particles. But we also found that we get this mysterious peak when we did that, which is highlighted with the arrow C. And basically that was a sort of again, a more of a complexing of the RNA, so backfolding, which gave us a high molecular weight peak. And by adding formamide to your sample in your extraction, that again broke down this complex and removed this high weight complex. So the formamide aids denaturing of the RNA in the extraction, and again removed this sort of ghost peak we saw at which is labeled C. So that's basically what we do now with mRNA, is we tend to have formamide to help with denaturing in mRNA. And again, there's application notes on the SCIEX website which go for this in more detail. So again, it's a very simple sample prep, right? You take your LNP sample, You extract it using Triton X-Reformid, you label it with cyber green, and then you analyze it using LIF, laser-induced fluorescence detection. But again, looking at this, and this is using an MC3 lipid nanoparticle, and also an SM102 lipid nanoparticle. These coatings, by the way, are related to the ionizable lipid used to build the LNPs. You can see we get highly reproducible results in both, which again allows us to do calculate the impurities present in the samples, which are the minor peak, which is migrating before the main peak. Which is around about, say, if you look at the MC3, it's around about 16 minutes. And then if we look at the size segmentation, you have to use an RNA ladder here. But if you compare it with an RNA ladder, we get very good size estimations when we calibrate the size based on the ladder. And you can see here we get only less than a 1% or about a 1% error in sizing when we actually do a size estimate, the RNA product, and we also get an estimate of the impurity as the size of the impurity as well, which gives us an idea what sort of clippings happened to produce that impurity. Another area we can use CE4 in this space is looking at poly-A tail characterization. And this is with single nucleotide resolution. So again, we sort of clip off the poly-A tail, and then we look to see how it differs between products. In this case, we use RNase T1 and oligo DTBs to sort of clip off the poly-A tail, and then we look at the lengths you see. Excuse me. And you can see that the poly-A tail in this case is 120 to 120, whereas the major peaks. This can tell you differences in production if the poly-A tail is changing. And again, in this case, we've gone back to using the 100R kit because it's around about the 100 base pair length. You want to get here a single base pair resolution and about that size. So we go back to the oligo kit, or the single-stranded kit. This really helps us to give sort of length determination of poly-A. And you can see here we see a slight shift between these two, and we see 120 mer on the red, and say the shift is about 121 mer on the blue trace. So there's a slight shift in profiles between the two, but it really helps us to give us a determination of the poly-A tail, and this is something which is again a request from the industry. And again, this single nucleotide resolution really helps to give us a good estimation of the size you get of your poly-A tail length. And again, this, if you look at reproducibility, it's highly reproducible, and this just shows you the example of separations you see off a number of different injections. And just to give an idea, the sort of sensitivity you get and the response you get from running this type of assay on CE is pretty linear. So you could use this to give an estimation of your titer, your level of RNA present in your product. And we've done this to give you sort of a genome titer of these LNPs. And this slide here just shows you sort of a curve of different concentrations which have been analyzed using our Biophase 8800 system. Again, as mentioned, it's linear, and this just shows the linearity of that curve I just previously showed in the previous slide. So it's a linear response. I also mentioned earlier on that you could apply this same sort of approach to single-stranded DNA samples. Single-stranded DNA is really also in AAV or viral vector is also an area of development of biopharmaceutical drugs. When you're using AAVs, they're normally used to treat diseases. If you use an AE or adenovirus, it would be used to treat, it would be used as a vaccine, for example, to help to treat COVID. But as I mentioned, this is also this sort of approach also been used to analyze single-stranded DNA. We call it again genome integrity analysis because we're basically using here to help to profile impurities. This just shows, one slide shows an example we've applied to several different adeno-associated viruses, particles or AAVs, going from serotype 2 up to 9, so 4 different serotypes. The sizing here is a bit more difficult to RNA in that in this case we use an RNA ladder to compare sizes against, and really you want to use a single-stranded DNA ladder to get a slightly more accurate sizing, and we're continuing to work in this space to develop accurate sizing for single-stranded DNA by CE. But again, we can use CE because it's a linear response to help us give us an idea of genome concentration in our sample. In viral vectors, of course, your genetic material is no longer surrounded by a lipid nanoparticle, it's surrounded by a viral particle. And often you get empty viral particles as well as full ones. So understanding the genome content and the level of intact genome present in your sample is important because that's the potency of your sample. And again, this response is linear. And if you use, again, we're using fluorescence detections. Again, we're using classically cyber green and cyber gold again, even though it's DNA is single-stranded DNA in this case. So we can use the same sort of dyeing mechanism. We can get down to e to the 10 particle concentrations, and allows us to calculate the amount of genome present in the sample, as shown in this slide. So for the last section, I want to discuss how we apply CE now to look at sizing and analysis of plasmids and DNA. So why are plasmids important? Well, plasmids are actually used in the manufacture of messenger RNA and viral particles. So they're a really important step in the manufacturing of some of these biopharmaceuticals. Plasmids or pDNA, as I mentioned, is an integral part of manufacturing nucleic acids. But they can contain topological variants, and these are sort of shape variants, and these include things like supercoiled, open circular, and linear isoforms. And these characterizations of these levels of these variants is important, and FDA recommends that actually plasmids contain the majority of the supercoiled variants to be used in manufacturing of viral vectors, for example. And again, capillary gel electrophoresis can be used to look at these shape variants or topological variants. Here now we're dealing with double-stranded genetic material or DNA. So we have to shift and use a different dye. In this case, we use a lift-floor enhanced CE dye, and this dye is an intercalating dye. It actually sits in between the double helix, but again, we're using a dye to label the genetic material and using laser-induced fluorescence detection. But when you do that, you can actually profile plasmids and look at the topological variants. And this shows you just an example where we picked up and looked at supercoiled versus open circular versus clipped variants. And you can see here the supercoiled is well separated from the open circular variant. Excuse me. And this is a plasmid of around about 5 kilobases in size, so 5 kb. But we've actually applied this separation to several different sizes of plasmids. But again, as I mentioned, you can apply it to not only look at supercoiled and open circular, but you can also pick up linear plasmids as well. So it's really good for testing your plasmid purity. And again, it's used in CDMO manufacturing now to look at the plasmids which are going to be used for viral vector manufacturing. And actually, there are several application notes and webinars in this space, and we've actually gone up to looking at much larger plasmids, including a 10 kilobase plasmid on the P800+ system. And what we do then is we basically use a double-stranded DNA gel, and we dilute it when we go to the larger plasmid lengths. We're using tris borates EDTA buffer or TBE buffer. And then we vary the injection conditions using electrokinetic or pressure to do the analysis. But it's really a sensitive approach for understanding your plasmid purity. Again, using this enhanced dye, if you want to look at this double-stranded DNA 1000 test kit, you can actually apply this to profile sizes of DNA, not just the topological shape differences you see in plasmids. And here we can see different sizes of DNA going up to 50,000 base pairs in length. And it's really quite a sensitive technique. And I said the LOD is down at 0.79 nanograms per mil with nearly a 3-order dynamic range. And again, as with all the other methods, CE is very reproducible, and this is shown here in this slide how reproducible this assay is. And this has been run on a P100+ system. So at the end of this presentation, I'd just like to summarize some of the key points. You can really profile, use CE to profiling small interfering RNA and small RNA. In these small RNA, the concentration is higher, you tend to use UV detection, and we've used this to enable process analytical development. And if, for example, I showed that earlier case with non-tritillated species. CE is very reproducible and sensitive, especially if you move to using laser-induced fluorescence, and can be applied to separation and detection of larger guide RNA and prime editing guide RNA molecules by simply changing the gel chemistry and sample conditions. And moving to lift detection, as I mentioned, really enhances the sensitivity, and it allows it to be applied to CRISPR product characterization. Again, by moving to different gels, you can profile messenger RNA and use it to understand size-based impurities of your messenger RNA used in LNP drugs. Again, you're applying the same method. You can use it to understand size-based impurities of single-stranded DNA used in adeno-associated viral products. And again, by changing the gel again on the same system, you can use it to profile plasmid DNA or even longer double-stranded DNAs up to 15,000 base pairs using our double-stranded DNA kit. Most of these methods can be applied to both systems, both the multi-capillary biophase system and the single capillary P100+ system. And we've applied actually both the oligos and the messenger RNA methods to both systems. The plasmid DNA method is currently only available on the P100+ method system. But I say you can apply this technology across both platforms, generally speaking. At the end of this talk, I'd like to thank you for your attention today, and again, I'll be open up for questions. And thank you for attending the presentation today.